Hello and welcome to Bio40 Pre-Lecture 10. Let's get into the structure of the double helix and the consequences of that information. James Watson and Francis Crick were able to construct an accurate model of DNA consistent with the previously discussed information on nucleic acid chemistry, Erwin Chargaff's rules about the ratio of bases, and Rosalind Franklin's X-ray crystallography data. There are serious ethical issues about how they obtained that last bit of information, which we'll certainly discuss in class. Watson and Crick conceived of a double helix with the sugar-phosphate backbone facing outward and the bases pairing inside the helix. Two pyrimidines pairing would be narrower than the 20 angstrom diameter Franklin observed, and two purines pairing would be too wide. But a pyrimidine pairing with a purine would be just right. And if every thymine was paired with an adenine, and every cytosine with a guanine, you would get the proper diameter, and have the A to T and G to C ratios seen by Chargaff. In arranging these pairs, Watson and Crick saw that 2 hydrogen bonds should form between A's and T's, and 3 hydrogen bonds should form between G's and C's. These interactions allow the double helix to stay together without any covalent bonds between the two strands. Here's a double helix in all its glory. 2 right-handed helices with the sugar-phosphate backbone on the outside, and the bases paired and stacked flat in the middle. The distance between each base is 3.4 angstroms, explaining the 3.4 angstrom periodicity Franklin saw. And each turn of the helix has 10 bases, explaining the 34 angstrom period in Franklin's data. The backbone is connected by strong covalent bonds, while the two strands are connected by the weaker hydrogen bonds between the bases. You'll also notice there is uneven spacing between the strands. This creates what we call a major groove and a minor groove. Finally, another important aspect to note is that the strands run anti-parallel. That is, one strand runs from 5' to 3', while the other runs from 3' to 5'. On the lessons page, I've included another video that includes mostly the same information, but shown a little more dynamically. Here's another view of the anti-parallel orientation of the strands. You'll see the significance of this orientation in many of the applications that we'll discuss in the next several weeks. I said before that after DNA was confirmed as a genetic molecule, there was a race to determine the structure, and now we have it. But why was this such a big deal to everyone? What does the structure tell us? Well, we're going to spend the next month going through all the connections between the structure and the function of DNA. We're going to cover how information is stored in DNA, how the information is copied and transmitted through generations, the mechanisms that alter genetic information, which is key to making evolution possible, and how that information controls the expression of phenotypes. Let's start with the nature of genetic information. The information content of DNA is found in the specific series of bases. Just as a string of zeros and ones contains the necessary information for a computer to function, the chain of A's, C's, G's, and T's along a DNA strand contain the information for a cell to function. Typically, to read this information, the double helix is unwound and the base pairs separate, which is relatively easy to do since the strands are not connected by covalent bonds. Oftentimes, this reading is done by using the DNA as a template to synthesize a new piece of DNA or RNA, and we'll get into how these RNA copies are read in a later lecture. Not all ways of reading DNA require unwinding, though. For instance, there are proteins that bind double-stranded DNA and control whether nearby genes can be expressed. Now, for the next couple lectures, we're going to focus on the second of the questions I presented earlier: how genetic information is copied so it can be passed to future generations. That is, how is DNA replicated? When Watson and Crick published their paper on the structure of DNA, they closed with what may be one of the great understatements of science literature. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. What they were referring to is called semi-conservative replication. In this model, the original molecule unwinds, and each strand serves as a template for a new double helix. Base pairing rules means a T on the template will always be paired with an A, A's with T's, G's with C's, and C's with G's. Each strand independently has all the information required to make a new double helix. So you get 2 identical daughter helices, each with 1 parent strand and 1 newly synthesized strand. While this model certainly sounds promising, we still need evidence to back it. After all, there are other possible, if less elegant, models of replication. In conservative replication, the parent DNA remains intact, and a copy is created using two newly synthesized strands. And in dispersive replication, the parent DNA gets split between the daughter helices, but in multiple pieces. We'll talk about the experiment that allowed us to distinguish between these models in class. But for now, just know that semi-conservative replication won the day, and let's dig into the details of how it works. DNA replication requires several factors. Obviously, you need the information you're copying, the parent strands. You also need the building blocks to create the new strands, the nitrogenous bases, sugars, and phosphates. Building a new DNA molecule takes a lot of energy, so you'll also need an energy source. Finally, you'll need enzymes to carry out all the chemical reactions that go into building new strands of DNA, and there are a lot of them. The most important of the enzymes are DNA polymerases. These are enzymes responsible for extending the new DNA chain with the proper base. The substrates for DNA polymerase are deoxynucleoside triphosphates, and there are different ones for each base: DATP, DCTP, DGTP, and DTTP. Collectively, they are called DNTPs. You'll see that they resemble the nucleotides we talked about earlier, but they have 3 phosphates attached to the 5' carbon instead of just 1. These triphosphate groups are highly energetic and provide the energy for the process. DNA polymerases take these DNTPs and attach them to the hydroxyl on the 3' carbon at the end of the previous nucleotide in the chain, making sure that the new base pairs correctly with the base on the template strand. Here's an example of how the process works. On the right is a template strand, and on the left is a new strand being synthesized. This thymine needs to be paired with an adenine. So the polymerase makes sure a DATP molecule is brought in, and it catalyzes the formation of a covalent bond between the first phosphate of the DATP and the 3' hydroxyl of the previous nucleotide, releasing the other 2 phosphates. Breaking this bond between the phosphates releases the energy required for the process. Now adenine is the end of the chain, and the process starts again, this time adding a DCTP to pair with the template guanine. Because the new base is always added to the 3' end, replication is said to occur in the 5' to 3' direction. Since DNA polymerase can only add DNTPs to existing 3' hydroxyls, it can extend a DNA chain, but not start a new one. So how does the new chain begin? Well, RNA polymerases can synthesize RNA strands without a pre-existing 3' hydroxyl. So a special RNA polymerase called a primase creates a short RNA chain, or primer, where synthesis begins. And DNA polymerase can use the 3' hydroxyl of the RNA primer to extend a DNA chain. Just as the DNTPs provide the energy for DNA synthesis, RNA building blocks, or NTPs, provide the energy to synthesize the primers. There's one more major consequence that comes from DNA polymerases only being able to add bases to 3' hydroxyls. Here's a very simple diagram of a replication fork. A spot where double-stranded DNA has split into 2 single strands to serve as templates for replication. The DNA has just started unwinding at the bottom, and the unwinding continues upward. A primer is made at the end of one strand, and the DNA polymerase extends in the 3' direction, moving toward the fork. Another primer is made on the other strand, and the DNA polymerase extends it towards the end of the DNA. Now let's move ahead in time. The fork has moved upward, and we want to continue copying the newly exposed portions of the strands. This is simple for the left strand. The DNA polymerase just keeps going in the 3' direction. But what about this gap? To fill that in, the process needs to start over. Primase needs to make a new primer at the fork, and the DNA polymerase extends it until it reaches the fragment of DNA that had just been synthesized. And this process repeats itself over and over again as a replication fork proceeds. The strand where synthesis is continuous is called the leading strand, and the discontinuous strand is the lagging strand. The different pieces of DNA that make up the lagging strand are called Okazaki fragments. In class, we'll talk about an experiment that demonstrated the existence of Okazaki fragments. Here's what the process looks like on a somewhat more detailed replication fork. Because one strand synthesis is continuous and the other is not, the whole process is called semi-discontinuous synthesis. Now we don't want our daughter strand to be a mix of RNA primers and regular DNA, So certain DNA polymerases have 5' to 3' exonuclease activity. That is, they can chew away nucleotides in the 5' to 3' direction. Such polymerases remove the RNA primers and replace them with DNA. But the polymerase cannot join Okazaki fragments together. At this point, the adjacent Okazaki fragments are not covalently bound. So an enzyme called DNA ligase forms a phosphodiester bond between the adjacent fragments, sealing the space in the backbone. This process requires energy in the form of ATP, a very common energy storage molecule that you'll see used in all sorts of biological processes. In addition to the 5' to 3' exonuclease activity, many DNA polymerases also have 3' to 5' exonuclease activity. This allows them to remove and replace bases they just added, which is important for correcting errors that occur in the process of replication. But even with this proofreading ability, replication isn't perfect. There are a few more important proteins worth bringing up before we call it a day. An initiator protein kicks off the whole process by binding the starting site or origin of replication. It recruits DNA helicase, which does the job of creating the replication fork by unwinding the DNA and separating the strands. This is another step in replication that requires energy in the form of ATP. Now sometimes DNA can become overwound and forms what we call supercoils, which replication forks can't move past. So an enzyme called topoisomerase, not shown here, moves ahead of the replication fork, relaxing supercoils. This also requires energy in the form of ATP. Finally, we have single-stranded binding proteins, which surprisingly enough are proteins that bind single-stranded DNA. They prevent the strands from coming back together, or annealing, before a new strand is synthesized. This concludes our overview of the pieces of DNA replication. Don't worry if you're still confused at this point. This is one of the more complicated processes we discuss in this class. The next lecture will begin by reviewing this information, but this time going in chronological order. We'll also get into some of the specifics about how replication differs in eukaryotes and prokaryotes.